Chapter 28, Invertebrates. When asked, most people will say that a type of vertebrate is their favorite animal. However, vertebrates only make up 3% of all animals. Although they are more well understood due to their similarities to us, as well as interest from the scientific community. Invertebrates include all animal phyla, including our phylum, Chordata. Vertebrates make up just a single subphyla of Chordata. So while invertebrates are more of a superficial grouping of anything that does not have a backbone, vertebrates are an actual monophyletic clade that includes all animals that have a backbone. This was a photo I took when I visited the Pacific Coast in northwest Washington for the first time. As you can see, this rock in the tidal zone consists of many different types of invertebrates, including echinoderms like that sea star, barnacles, which are part of arthropods, and sea anemones, which are part of phylum Cnidaria. Well, let's get started with the most ancestral types of animals. So the most ancestral of all animals belong in the clade Parazoa, which means beside the animals. Parazoans include Phylum Placozoa, with only one species, Trichoplax adherens, which resemble a multicellular amoeba. The other phylum is Phylum Porifera, also known as the sponges. Phylum Periphera is considered the most ancestral of animals because they lack true tissues. Functionally, the cells of Phylum Periphera do act as tissues, but they're not true tissues because they don't derive from those embryonic germ layers such as endoderm, ectoderm, and mesoderm. Sponges don't possess them. Like all animals, they are multicellular with internal digestion, and they move at some point in their lifetime. The only time sponges move is during the larval stage, where they swim and eventually plant themselves to the substrate. Sponges are essentially water filtration systems, drawing in water for feeding and gas exchange and excreting water with waste and carbon dioxide. The various canals and chambers help to distribute this water to all parts of the sponge's body. Thus far, 5,000 species of sponges have been identified in different aquatic environments worldwide, but there are likely much more. Sponges vary in morphology, but the simplest body plan is a large irregular cylinder with a large central cavity called the spongiocele. Water enters through the various pores, also known as ostia, and are expelled at a large opening called the osculum. Now please note that some sponges have elaborate labyrinths of chambers and canals that may not be represented by this model, but the path of water is still the same, entering through the ostia, going through the spongiocele, exiting through the osculum. And again, the whole point is their body plan is to act as one water filtration device in which the sponge collects food from suspended particles in the water. So sponges lack true tissue because true tissues derive from embryological tissues. However, they have functional tissues, meaning that the cells act tissue-like by working together. Cells of the sponge include pinacocytes, which act like epidermal tissue on the outside of the sponge, and emporocytes, which are specialized cells that regulate water flow into the sponge. The spongy feel is the result of structural cells that make up the mesohyl, which contains collagen fibers for pliability. Within mesohyl tissue lies amoebocytes, named due to their amoeba-like appearance. They help build the mesohyl and skeletal tissue called spicules. Amoebocytes have a variety of different functions, but move within the mesohyl. Nutrient delivery from choanocytes to other cells. Sponge spicules are mostly used for structural support, but also defense against predators. They're glassy-like and very sharp, which prevents a lot of predators from trying to consume sponges. One exception is sea turtles. The feeding chamber of sponges makes up the internal lining of the spongiocele and are made up of choanocytes, which bring in microscopic food particles. The flagellum of each choanocyte functions in drawing water with food particles in. These particles then fuse across the cell and into the mesohyl. Choanocytes can also be used as sperm cells for sexual reproduction. And if that name sounds familiar, it's because the choanocytes look a lot like choanoflagellates, which is why we consider animals to be part of the supergroup of Pistaconta. So sponges lack complex body systems. However, they do perform internal digestion by trapping food particles through filtration. Food gets trapped in water and enters the sponge ostia, and the water leaves the osculum on top. Food particles smaller than 0.5 microns, such as bacteria, are trapped by the choanocytes and ingested by phagocytosis. Larger particles are those too big to pass through the ostia and can be phagocytosized by the sponge surface pinacocytes. Amoebocytes can deliver food particles to various cells in the mesohyl. Afterwards, each cell completes the digestion of food particles. Gas exchange, circulation, and excretion of waste are all performed via diffusion between the cells that line the opening within the sponge and water. Some sponges have endosymbiont green algae and/or cyanobacteria and receive glucose from these partnerships. Some sponges are actually predatory and feed on small animals trapped in sticky threads or hooked spicules. Thus far, we have not found any sponges with a nervous system in this group, but intercellular communication helps to regulate activity of the whole sponge. Sponges reproduce sexually and asexually. Asexual reproduction methods include fragmentation, where a piece of the sponge breaks off and settles on a new substrate, and a new sponge grows from this. Budding happens, where a genetically identical outgrowth from the parent is formed and detaches to form a new sponge. In freshwater sponges, gammules are produced, which act very similar to cysts in protists, in which they can withstand harsh conditions and form into a sponge once times are better. Sexual reproduction occurs when amoebocytes differentiate into oocytes and retained in the spongocoel, and choanocytes differentiate into sperm cells and are ejected out of the osculum of the sponge and moved by water currents. Eventually, the sperm of one individual makes it to the egg of another. Sponges are monoecious, meaning that both gametes are made from the same individual. Although to reduce self-fertilization, some sponges will switch from egg to sperm production at different times. Sponge larvae develop early in the parent sponge and then are released via the osculum, where they swim to an appropriate location and plant themselves into the substrate. For the most part, sponges are sessile, but sponge cells can creep together along a substrate. In our next video, we'll be looking at Phylum Nidaria.